Okay, so we are going to dive into plants and talk about the evolutionary pathway from plants in water to adapting on life on land, basically. So I combined chapter 30 and 31 with the guided notes. So this is about seedless plants, plants that do not produce seeds. And then hopefully tomorrow we will start seed plants, but I'm not exactly sure how far we'll get. So here we go. Origin of land plants. We're going to recap a little bit about what we believe the phylogenetic tree, or I don't know, evolutionary tree shows with origin of plants. So yeah. So we know land plants evolved from freshwater algae. It is believed that a single species, one single species is responsible for all the land plants on Earth. So some type of freshwater algae. If you remember from a previous chapter, how they talked about green algae, how it's split up into two major clades, the chlorophytes and the charophytes, and how those chlorophytes never made it to land, and the charophytes did. And from the charophytes, we, have land plants. Oh, shoots. Hold on. I need to fix something on this PowerPoint slide because I have the wrong...
You remember to record?
Yep, I'm recording. Look at that. It's going to be, the mistake's going to be in here. I'm just going to fix it real quick. Okay. Sorry. So the chlorophytes, they were types of protists, never made it to land. The charophytes made it to land. And then from the charophytes, land plants stemmed from them. And so both charophytes and land plants are categorized underneath another clade called the streptophytes. So this was mentioned in a previous chapter. All of these, actually, this entire slide has been mentioned in a previous chapter. So the land plants, they're multicellular. They have haploid and diploid states. So we're going to talk about the haplodiplontic life cycle, or alternation of generations, how plants go from a haploid state to a diploid state, and the life cycle of plants. All right, can I move on? Okay, so this is where we've gone. We talked about protists, but they're at red algae, chlorophytes, which are the green algae, and then the charophytes, which turn into the land plants. So we are going to talk about the bryophytes, liverworts, mosses, hornworts, the lycophytes, and hopefully finish off with ferns. And then tomorrow we'll talk about the seed plants. So here we go. So green algae was confined to a watery environment, a domain, if you will, and it was necessary to have water for reproduction because the sperm would swim to the egg. Not only that, water gives you pretty good structural support, buoyancy, and since you're submerged in water, you don't have to worry about water loss. However, if you're going to adapt to that terrestrial life, you got to overcome some things. First off, desiccation, drying out. So how do you protect yourself from drying out? Well, plants kind of developed this cuticle, this waxy substance on the outside that slows water loss and still allow light to come through. But then if you're covering your entire body, your plant body, with cuticle, well then how do you get gas exchange for cellular respiration and photosynthesis? You got to take in CO2, you got to release oxygen. So now plants have stomatas, tiny little openings on the underside of the leaves made up of two guard cells that open and close like mouths, and they allow gas diffusion. Another thing is, as a plant size increases, you got to be able to transport water and other materials. So we'll talk about the development of a vascular system, which is composed of two types of tissues, or two types of specialized cells, xylem and phloem. With protists and green algae, their main life cycle is haploid. They're usually in a haploid state. But now with life on land, we're going to see cells that are diploid. And the purpose of that is to minimize mutations, due to high rates of exposure to UV since you're on land. And so all land plants do shift from a haploid to a diploid state and back to it again. They alternate between those two. So of these listed, adaptations that are important, I would say that number 4, protection from UV intensity, and then water efficiency, which kind of ties into 1. So those are kind of like the 2 most important. How do you prevent water loss, and then how do you protect yourself from UV/drying out? Okay, so the haplodiplontic cycle, probably saying that wrong, produces the alternation of generation, going from haploid to diploid states. Now, animals are diplonic. That means that only the diploid stage, the full set of chromosome numbers, is multicellular. So we have meiosis that produces our haploid gametes, but these gametes never go through mitosis to become multicellular. They remain like individual cells, and then they'll fuse and become diploid. So there is no multicellular haploid generation in animals. Plants, on the other hand, do have multicellular haploid states. So haplodiplontic, maybe that's how you say that. So they have multicellular haploid and diploid stages. That means that mitosis occurs after fertilization and meiosis. So if we have fertilization, 2 gametes meet, it's diploid. That cell goes through multiple rounds of division to become multicellular. And then that diploid part of the plant will undergo meiosis to produce a haploid gamete or spore. And then that itself will go through mitosis and become multicellular. So the multicellular diploid state, we call that a sporophyte. It's going to produce spores. And on the flip side, the multicellular haploid state is called the gametophyte because it's going to produce gametes. So how a sporophyte produces spores? In a specialized structure called the sporangia. There's going to be tons of vocab here. Meiosis occurs in diploid spore mother cells. So they were diploid, they go through meiosis, now they become haploid. And then they'll be released and go through mitosis to produce a gametophyte. Then in the gametophyte, gametes are produced through mitosis, and then those gametes are released and they'll fuse and form a zygote, which is diploid, and produce the sporophyte structure. And then we just kind of repeat. So we alternate between a diploid and haploid state. All right. So here's a diagram that shows this haplodiplontic state. We'll start with fertilization. So here we have gametes. They fuse in fertilization, become a zygote. So we're diploid. Okay? That diploid zygote will go through multiple rounds of mitosis to become multicellular, and now we have a sporophyte. There's a structure on the sporophyte called the sporangia, where inside we have some spore mother cells that are diploid. They will go through meiosis and produce gametes, or haploid spores. I probably should use the word spores here. These spores will get released. We'll talk about how they get released. Some are water-based, some are wind-based. But when they land or find a suitable environment, they will go through multiple rounds of mitosis to become a gametophyte, and that's the multicellular haploid state. And then they will go through more rounds of mitosis to make gametes that will be released and fused. Now, inside the gametophyte, there are structures called the gametangia, and that is where gametes are made, because you can have egg, like female gametes, and male gametes. So the antheridia is the male gamete, and the archegona is the female gamete. So this is just a generalized. You're going to see various diagrams with the moss life cycle, the fern life cycle, the lycophyte life cycle. Yeah, so this is the book that this is the diagram that's in your book. It's very similar. You know, here's our, we'll start a fertilization, egg, sperm, meat, zygote, go through multiple rounds of mitosis. I just, hate that they don't mention mitosis or meiosis in this one. So you have mitosis where it gets larger and we produce more cells to a sporophyte individual inside the sporangia, or the spore mother cells that go through meiosis to produce spores. Spores get released, they land somewhere, they go through more rounds of mitosis to form a gametophyte, and then to make those gametes in the archegonia and antheridia, you form these gametes. Yeah. Different colored slides, you don't need to write anything. So I wanted to show you a picture of what this female and male gametophyte parts are called. So the female is called the gametangia, or the archegonium, and you can see it right here, and that there's the egg on the inside. Okay, so the structure is the archegonium, and inside is where the egg is. The egg does not move. Okay, sperm has to swim to it. So here's the male side, the antheridia. Okay, and they can be released through wind or water, but as you can see here, the sperm is flaginated. So which method, air or water, do you think it's going to use? Air or water? Like, if it's flaginated? Water. Okay. A lot of times they just need a raindrop, and they can swim great distances. So you can see that the antheridium is just filled with flaginated sperm being ready to release, and usually this capsule will bust open when it desiccates or dries, and I'll show you a really cool video on that hopefully today. I am going to post this in Google Classroom when I get a chance, but this is basically all the vocab terms you need to know, conveniently on one slide for you. Okay, back to notes. So you're going to see that as we go through the evolutionary history of plants, going from simple to, let's just say, most complex, you're going to see the size of these haploid and diploid generations vary. We're going to start off where the life cycle of a plant is mostly haploid. And as we progress, the haploid state gets smaller and smaller and smaller, and the diploid generation kind of takes over. So when I talk about mosses and ferns versus seed plants, the haploid generation consumes more than 50% of the life cycle. When we look at sizes of the gametophyte versus the sporophyte, in the mosses and ferns, I'm just going to say mosses, the gametophyte is the biggest and the sporophyte is small. And not only that, it's attached and dependent on the gametophyte for survival. But in the ferns, you see a flip where the gametophyte is photosynthetic, but it's small, and the sporophyte is large. And then tomorrow when we get to seed plants, you're going to see that the gametophyte is microscopic. So you can see that as we progress, the gametophyte just gets smaller and smaller and smaller. So over here I have a reduction in gametangia, which is the gametophyte, as we progress. last slide for the section. Some other things. In our simpler plants, like the mosses, egg is stationary, sperm has to swim to it. But then tomorrow when we get to our seed plants, you're going to see that the gametophyte is only made up of a few cells, it's microscopic, and sperm no longer need to swim. So we're going to throw in some other mechanisms for the sperm to get to the egg, the archegonium. Review questions, and there's a lot of them. I could have added a lot more, but I was like, okay, this is already getting long. The plant life cycle is categorized as A, haplodiplontic. Number 2, in plant life cycles, which of the following is correct? Make sure you go through all the options. It's kind of tricky. Yes, it is D. It's not sporophyte to spores because this would have to be meiosis. Spores to meiosis to gametophytes, no, that would be mitosis. Gametophyte to meiosis, nope, it's already haploid, so we would just do mitosis here. Zygote, that's diploid, to sporophyte, that's diploid. Diploid becomes spores, which are haploid through meiosis, so it's D. So, yeah. And then this one, egg to zygote to mitosis, that would, and then to spores, you'd need meiosis in here. So, all right, number 3. Which of the following plant structures is not matched to its correct function? I don't know if I mentioned tracheids, but it's D. All of them are matched correctly. Okay. So tracheids are like xylem and phloem, and so we'll talk about them in depth here, hopefully today. Number 4, before aquatic plants can move onto land, they had to D, all of the above. Develop a way to absorb minerals. We'll talk about the evolution of roots, find ways to conserve water, cuticles, stomata. Develop a way to reproduce on land, and you'll see the various ways they do that. Next question, what is the first member of a gametophyte generation? First haploid state. What? C, that's diploid. Gametophyte's haploid. What? It is B, a spore. Okay. All the other ones, oh, I guess sperm, but we haven't talked about sperm. There's no sperm here, it's spore. Okay. Select the most important adaptation that was required of plants to survive on land. It is E. Yeah. Water management and mutation protection. True or false? The evolutionary trend in land plants is to reduce the size of the gametophyte and increase the size of the sporophyte.
True.
That is true. And the final one, select the plants from which originated the entire terrestrial plant lineage from mosses through the flowering plants. There's only one species. One species of freshwater green algae. Okay. All right, moving on to bryophytes. So I'm going to talk about some basic adaptations of bryophytes, which are mosses and liverworts and hornworts, and how those 3 clades are different from each other. But bryophytes, they were probably the first land plants, and they were probably the only plants on Earth for like the first 100 million years. Yeah. And so from the bryophytes, then all the other plants kind of derived from. So some major features of bryophytes. Okay, first off, no vascular tissue. And I think I mentioned this in my notes. They're non-tracheal. Yes, you'll see it on the next slide. There's no vascular tissue. This is the Sporophytes, okay? And this is the Gametophyte. And you can see that the Gametophyte is a lot larger than the Sporophyte. So I talked about the reduction of gametangia. Okay, well here the Gametophytes are big. They're dominant. Sporophyte is short-lived. It's unbranched. A lot of times it's parasitic, which means it's dependent on the Gametophyte for everything. You can see it's attached to the Gametophyte. It needs the Gametophyte in order to, carry out its functions. All right, so general characteristics. Closest living relatives to the first land plants. They are non-tracheophytes. They don't have any tracheids. They don't have any vessel elements. They don't have any xylem or phloem. So these guys, because they don't have a great way to move water, they're still tied to water. And some of them formed mycorrhizal associations with fungi to help them take in water. It's made up of 3 clades: the liverworts, the hornworts, and the mosses. The liverworts are probably the ancient phylum, as you can see here. It's the first branch in point. And then we have the mosses and the hornworts. Now this is kind of interesting. In the old edition of our book, hornworts was listed here and mosses were here. So just recently, within the last 10 years, they've come across DNA evidence that supports that hornworts are actually closer to tracheophytes than mosses. So I think that's kind of cool. But you're going to see that, yes, we are still tied to water. You'll see with fertilization how sperm are flaginated and how they have to swim to the egg here. So, all right, the Gametophyte. Oh, sorry, I can go back. I'm sorry. Okay. So the bryophytes, their gametophyte is photosynthetic. It's the dominant life cycle part. But we are still small. Mosses are small in general. Now they don't have tracheids, but they do have cells that can carry out the job of the tracheids. Simple conducting cells. So water and nutrients kind of move passively throughout the organism, and that's why we're still tied to water here. Osmosis, diffusion. The egg and sperm unite, and they'll form the sporophyte that grows from the gametophytes. So the sporophyte is dependent on the gametophyte, grows off of the gametophytes. Spores are released. They can be carried by winds. They can also be carried by raindrops. And I should probably add raindrops there. But bryophytes are found in moist places. So as you can see, we can kind of trace our way back to that aquatic life. All right, so the next couple slides, I'm going to go through each of the clades, starting with liverworts, then I'll go to mosses, and then I'll go to hornworts.
Wait, so where did you want the raindrop?
Carried by wind/rain drop. Your book says wind, and I'm like, yeah, I thought it was rain. I don't know. So. Oh, unless it's talking about spores. Okay, maybe. I don't know. Never mind. I am tired. Sleep deprived. So, spores are the haploid cells. They land and germinate into gametophytes. So maybe they are carried by wind. And during the process of fertilization, they need water. That's probably where I'm confused. Yeah. All right, liverworts. Some common characteristics. Their gametophyte is flat, not upright. Okay, so flat. And you can look at it, it literally looks like a lobe of liver, okay? However, 20% of liverworts look like liver, the other 80% don't. So it's kind of funny in a way. The gametophyte is kind of anchored to the soil with these single-celled rhizoids. They're like roots, but they are not true roots. They're made up of single cells, so it's not an organ. Okay? So they're not roots. Liverworts also have a stomata-like structure. Basically, it's just a pore, an opening, and it's fixed open all the time. There's no close-in with guard cells, just open all the time. Okay, so let's take a look at just a quick life cycle of liverworts here. Here's the male, antheridia. Okay, it's going to produce male spores or sperm, if you will. And then here's the female. So when it rains, it splashes, and the sperm basically jump in that raindrop. So here's the raindrop. You can see that the flaginated sperm get carried up with microsplashes, and they hopefully land on an archegonia head of the female part, and they will swim underneath to the archegonia underneath the archegonia head. Okay, so here's the male gametophyte, the female gametophyte. Through raindrops, they make the jump and swim. All right, mosses. All right, their gametophyte is actually one cell layer thick, has these leaf-like projections. They're not true leaves because it's haploid and it lacks vascular tissue. But they are photosynthetic. And again, we see that they are anchored with rhizoids, not true roots, but similar to roots. These guys don't have a vascular tissue either, so water moves passively from high concentrations to low concentrations. They don't have a stomata. The only part of a moss that has a stomata is the capsule on the sporophyte. I think it's the sporophyte. Yes, it's the sporophyte. Okay, so here are the spores, and you can see that we have male and female spores, and they will germinate. So I found a microscopic picture of them actually going through germination to form a young gametophyte, if you will. So here's the female gametophyte, here's the male gametophyte. If you take a look at this picture, the ones that are yellowish, those are the male, and those are the female. And so when it rains, hopefully those microscopic splashes will land on the female archegonium so they can swim to the egg. So here's the antheridial head. You can see it's loaded up with flaginated sperm here. And so when the rain comes, it'll kind of displace them and lift them up and head to the female part. So here's the capsule that I was talking about where you only find the stomata. So the spores get released. They germinate into the gametophytes. Then rain will occur. Okay, sperm meets egg in fertilization, and now we are diploid. So the red colored arrows here are diploid. So we have a zygote to an embryo to a young sporophyte. And you can see that the sporophyte is growing off of the gametophytes, and it produces this capsule. This is the sporophyte, this is the gametophyte down here. So this is the sporophytes. And then inside the capsule, it will go through meiosis. So there's meiosis, the spore mother cells produce the spores, and the spores get released. So, yeah, I believe you have a diagram or a worksheet that you have to answer regarding the moss reproduction life cycle here. But your book also has a moss life cycle diagram. I found a picture also of still life, okay, pictures of spores being released, which is so cool. So that's the sporophyte, that's the gametophyte. There's the little capsule on the top. And when it dries, I mean, they just pop the top off, and you can see the dispersion of the spores here. It's crazy. So, yeah. Okay, so where do you find moss? You can find them in the Arctic and the Antarctic due to poor soil. So they have that permafrost up there. You know, they only have like 6 inches to maybe a foot or 2 of soil. So you don't find any trees up there, but you do find lichens and moss. You can also find mosses in the tropics, and that's probably where you'll find the greatest diversity because of its moist environments. Moss is highly sensitive to air pollution, does not do well with pollutants. And another use of moss, it's very absorbent. It can actually absorb up to 25 times its weight. So you may find moss commercially in potting soils, and then also with the use of fuels. So. All right. Last clade with the bryophytes, hornworts. So they think hornworts are closely related to the tracheophytes, plants that have tracheids. Hornworts, there's only about 200 species worldwide. Their sporophyte is photosynthetic, but it still grows off of the gametophyte, and it has stomata. They also have a single large chloroplast in each cell, which is why they think hornworts are a sister group of tracheophytes. We see the chloroplast in each cell. We see a stomata, and you can see that it has guard cells. A lot of times hornworts will form a symbiotic relationship with cyanobacteria for nitrogen fixation. And just to give you a scale reference of how big these hornworts really are, there's a dime and there's the hornwort. So they're small. And I'm sure you can kind of guess why they are called hornworts, because the sporophyte is like a tapered horn. All right, review questions. In the Arctic, blank are the most abundant plants. Mosses. A characteristic feature of mosses, liverworts, hornworts is the presence of usually colorless projections called rhizoids, or rhizoids, I don't know how to say it. The phylum of plants that includes mosses is called C. Bryophyta. These moss structures consist of several cells that absorb water, but not nearly the volume of water that is absorbed by a vascular plant roots. Which structures am I talking about here?
B.
What?
B.
No. A. Rhizoids again. Again. Okay, tracheophytes. So they have a dominant sporophyte generation. Talk about what tracheids are, and then the evolution of roots, stems, and leaves in the tracheophytes. So remember, in an aquatic environment, you know, plants or green algae, we'll say early plants, cells were surrounded by water, minerals were dissolved, so it was easy to absorb in, as well as CO2 and oxygen. We have structural supports with buoyancy, unicellular reproductive structures that could swim in that environment, and all cells were photosynthetic. So you're going to see even more with the progression here. How do Land plants disperse their gametes in drier environments, so we're moving away from water. We're going to start cutting the ties. How do we protect the zygote and embryo from drying out? So you'll see the evolution of seeds tomorrow. How to take up water and nutrients from below ground. So instead of rhizoids, now we're actually going to develop roots. How to take up CO2 from the air, stomatas. How to resist gravity. We'll talk about the tracheids and vessel elements. And then move in water and nutrients long distances. I guess I should also mention cellulose and ligand as well. So some of these features are going to be, you're going to see them again when we go back to plants after animals. But ground tissue, we'll talk about parenchyma and sclerenchyma, the epidermis, the cuticle, and the stomatas, the vascular tissue, how that includes xylem and phloem, roots. I won't discuss much about lignin, but yeah, here we go. So the first tracheophyte belonged to the phylum Rhyniophyta. It appeared about 410 million years ago, but now it's extinct. We do have fossils of Cooksonia that give us insight into what they may have looked like or what they did. And we realized that they were probably very successful due to little competition because, let's face it, there wasn't a lot of land plants on land at that time. Another unique thing about the first tracheophytes, in this case Cooksonia, was that it's homosporous. It only made one spore. So this diagram over here is showing homosporous versus heterosporous. So with the homosporous, we get one spore. It produces a bisexual gamete that can produce both eggs and sperm. In some of the previous life cycles slides that we saw in this lecture, you saw that we had female gametophytes and male gametophytes, and the female gametophytes only produce eggs, and the male gametophyte only produces sperm. So that is called heterospores because they produce two different spores. So homosporous is bisexual. It can produce both eggs and sperm. But the major feature of tracheids is that our sporophyte is now becoming more dominant versus the gametophytes. All right, so first up, vascular tissue. So with the development of vascular tissue, we can transport substances long distances. This tissue is made up of two types of specialized cells that run throughout it. So it runs through the roots, the stems, and the leaves. They are called xylem and phloem. So xylem moves water and anything that's dissolved in that water, and it moves from the roots up through the stem to the leaves. So from the roots to the shoots. Phloem conducts sucrose, which is sugar, as well as hormones throughout the plants. So phloem, I think of food. Sugar is food. So phloem moves food. So in the next couple sections, you'll see us transition from tracheids, which are cells that conduct water and xylem tissue, to vessel elements, where we kind of throw in phloem and a few other specialized cells like sieve tubes, and there's another type of cell that I can't think of, but it happens in seed plants. So now that we have vascular tissues, our plants can grow in height as well as size. And another thing is that these vascular tissues are only found in the sporophyte, not the gametophytes. So the tracheophytes, you can include the 7 current phyla groups, but we've taken those 7 current phyla groups and we've grouped them down into 3 clades. So the lycophytes will get their own section, the pterophytes will get their own section, and the seed plants will get their own chapter, which is tomorrow, chapter 31, seed plants, which include gymnosperms and angiosperms. Okay, so with tracheophytes, we have roots, stems, and leaves. So let's talk a little bit about those stems. Stems evolved prior to roots, which is interesting. So plants grew tall before they kind of lay down a base, a foundation, if you will. So our early fossils, including the Cooksonia, they don't show roots or leaves in those fossil records. And so when you don't have roots, it kind of limits you in 2 ways. Roots anchor you to the ground, they keep you from falling over. And if you grow larger in size, then you have a larger surface area for more water loss. And so by developing an extensive root system, you can help with that huge increase in water demand. So with the tracheophytes, we see development of true roots. They provide structural support. They aid in transportation, water absorption, moving from the roots to the shoots, and they have evolved at least 2 separate times, if not more. But for sure, we know 2 separate times that roots have evolved. Some other things with the tracheophytes, leaves. In its evolutionary history, leaves have evolved more than once. With the development of leaves, you increase your surface area, you can take in more sunlight, enhance your photosynthetic capacity. So the next section is about the lycophytes. And the lycophytes have true leaves, but they're really, really small, and we call them lycophylls. And one thing about the lycophytes' leaves is that it has a single vascular strand or vein in each leaf. Whereas with true leaves, the Euphylls, which you find in ferns and seed plants, they have highly branched, complex vascular strands within each leaf. So lycophylls, they kind of have simple leaves, just that one vein that runs through it, the vascular tissue. And then finally, seeds. which is all about tomorrow. Highly resistant structures, they protect the plant embryo from stressors like drought and predation. They can be eaten by animals and not be destroyed. So the lycophytes and the pterophytes, which is section 5, I think, they lack seeds. So we're still discussing haploid spores today, but tomorrow is all about seeds. So another thing with seeds, seeds, they can be dispersed in many ways: wind, water, animals, and as a result, you increase your range that the species could live in, and you can also promote genetic diversity with it. So, all right, so that does it for section 30.3. Moving on to lycophytes. And I think this might be a good time to stop because the bell's about to ring, but I guess we'll continue off with lycophytes right here. So I'll slide it over this way. I just need to check.
Carter Martinek, please come down to the office. Carter Martinek, please come down to the office. Thank you.
So for those like ferns,